Thank you everybody for coming today. This is like a record week of seminars, I know, so I really appreciate your time. My name is Dr. Betsy Boughton. I'm the program director of agroecology. And do we have any new people in the room? Aaron.
We have Delana Rogers, our new seasonal research assistant. He just started this week.
Oh, great. Oh, welcome.
Here, hopefully
Okay, so I'm really happy today to introduce Abigail Schulken, or Abby as we call her. She's been in our lab since about October. Yeah, so it's been really fun to have Abby in the lab because she is probably the most passionate person about soils I have ever met. Never seen someone so excited, and it's very contagious, so I appreciate that. And Abby has also tried to spread that contagion through some of our education events with kids and master naturalists, and has helped with our education and outreach on soils, so it's been really great. Abby comes to us from Virginia Tech, and she got her bachelor's there in 2024. And she got a Bachelor of Science in Biology with a focus on ecology, evolution, and behavior, and 2 minors, one in civic agriculture and food systems and crop and soils. So she has done a lot of work in different food systems and has worked with World Wildlife Fund to do an internship on agri-food innovations. And she's done several research assistantships, one in the invasive plant ecology lab at Virginia Tech, and another focused on crop weed science at Virginia Tech. And she came to the ranch to sort of help Greg with his virtual fence project. So you got to work with that. And then she discovered the overseeding project at the ranch and worked very closely with Karen Rice David, who I'd like to give a shout out to because Karen really guided Abby throughout this project. And a big shout out to all of our agroecology lab for helping each other with all of our projects. And thank you all very much for that. So I will turn it over to Abby, and she's going to talk about Cover Your Grass, assessing overseeded mixture interactions with competition, herbivory, and soil phosphorus. So take it away.
Hi, everybody. Thank you for coming to my executive director seminar. I'm very excited to be considered for the position. No, I'm kidding. My name is Abigail Schulken, and this is my intern seminar entitled Cover Your Grass, Assessing Overseeded Mixture Interactions with Competition, Herbivory, and Soil Phosphorus. Okay, so it's not going, but I'm going to do the cursor thing that Laura told me about, and then it will. Okay, so you might be saying to yourself, what is overseeding? And that's a great question, and where we're going to start today. Overseeding is a best management practice that is most often used in areas where the pasture land forages experience a period of dormancy. And it's used it entails planting cover crops right into your pasture land. So it's not like you're not clearing the land, you're just putting them right in there. And it's used to get all these wonderful benefits that I would read to you, but I have so many slides that I'm going to skip for today. And you might also be saying, this sounds so familiar, but as Betsy pointed out, there's this current project happening at Buck Island Ranch that has to do with overseeding, where we want to connect these Florida Department of Agriculture goals to some of our Buck Island research goals to overseeding. And I got really interested in this project because, as Betsy pointed out, I'm a huge nerd about soil. It's the coolest. And this idea that we could connect this legacy phosphorus issue to soil water quality and to overseeding was super interesting to me. Okay, so this is a spodosol. And if you saw my prop coming in, this is also a spodosol over here. And spodosols are what we have at the ranch. That's the soil order that we see most of the time. Up here, the plant roots, and up here, or down here in the spodic layer is where this legacy phosphorus is. And legacy phosphorus refers to these historical additions of phosphorus to the topsoil at Buck Island Ranch before, like in the 1980s and before. And this inability of plants to take up all of this is like this fertilizer phosphorus, and so it leaches downward. Spodosols are actually known for their leaching capacity. Like they have a hard time holding nutrients up here, and so it just kind of leaches into the soil profile. And so as the water table rises and falls, going up and down, I love this little pointer thing. As the water table rises and falls, this phosphorus is moving around, and then eventually it's going to move out of the soil profile and into the watershed, where it becomes this huge water quality issue. So how do we fix this? That's one of the things that overseeding is trying to address by energizing with functional diversity. Basically, if you plant a lot of different plants with a lot of different root systems that function in very different ways, you're going to see a bunch of different benefits like occur. But all of these wonderful benefits are only possible if you can get your plants to establish. And over the last 3 years of the project, there's been minimal establishment at Buck Island Ranch. So why? These are kind of the 3 reasons we kind of came up with. The first is that Bahia grass is outcompeting these overseeded mixtures. This is an improved pasture, which is where Bahia grass is planted. It's filled with Florida's favorite forage, Paspalum notatum, which is just Bahia grass. And as you can see up in the top layer of soil, it's concentrating 65% of its root biomass. And it's spreading vegetatively, so it forms this very, very thick sod layer. And so if you're a tiny plant, you might have a really hard time germinating with this massive competitor just like hanging out with you. So that was one of the things that we thought might be holding it back. Another is herbivory, and I'd like to stress not by cows. We mean like deer, hogs, rabbits. I think they might be eating the overseeded species as they emerge. And then finally, we have environmental conditions. I mentioned that this overseeding is something that happens in places that experience periods of dormancy. And if you've spent any time in South Florida, which everybody in this room has, you know it doesn't get cold here. So we don't know if these Bahia grass plants are always going through a period of dormancy. So maybe this is just not right for South Florida. So to kind of address these questions and some of the soil phosphorus questions I had, came up with 3 hypotheses. 1, if competition is a strong factor, then when Bahia grass is cleared, there will be an increase in the growth and establishment of overseeded species. 2, if herbivory is a strong filter for overseeded species, then when herbivores are excluded, there will be an increase in the growth and establishment of overseeded species. And 3, if overseeded species can mitigate phosphorus, then there will be an increase in soil phosphorus storage capacity, thus a decrease in phosphorus loss from the soil. Okay, before we go any farther, let's establish where we are, especially for the people on Zoom. We are in Florida, and if we zoom in, we are in South Central Florida on a 10,500 acre working cattle ranch known as Buck Island Ranch. And if we zoom in a little bit farther, we arrive at pasture I, which is an improved pasture filled with Bahia grass. It's at the north end of the ranch, right off the ranch road. So like if you drove in anytime between December and March, you probably just saw me like wandering around out there. Yeah. And okay. For this project, we split pasture I into 3 different sections, but we only used 2 of them. In the middle, we planted our winter mix that had lesser amount of diversity. And this had crimson clover, daikon radishes, and turnips. And you can see their planting rates are listed below. And on the right-hand side of the pasture, we planted winter mix 4. I do not know what happened to winter mixes 2 and 3. I get this question all the time. So just maybe 2 and 3 don't exist. I don't know. But winter mix 4 had more diversity in it. And again, we had crimson clover, daikon radishes, and turnips, although they were planted at different rates. And we added oats, wheat, and hairy vetch to this mix. Triticale was also planted, but we saw no evidence that it germinated. So like for the purposes of this presentation, just forget that it exists. Okay, so how do we do this? Well, it all started with 1 acre of pasture I being disked. And for those of you unfamiliar with disking, it's a tillage practice that involves like chopping up the first 30 centimeters of soil, disturbing it, and completely clearing it of any vegetation. So in this case, we cleared it of all of this bahia grass. And we also picked a corresponding wood acre strip that we did not disc to serve as the control area. And then, so after operations disked this area, we conducted the first round of soil sampling. We took 16 samples from the cleared section and 16 samples from the bahia grass section for a total of 32 sample points. And at each of these sample points, we took samples from 4 different soil depths for a total of 128 samples of soil, which is so many soil samples to then dry and sieve and send off to college, aka the University of Florida Soil Testing Lab, where they did phosphorus testing for us. After we soil tested, we took the samples of the soil. Winter mix 1 and winter mix 4 were planted in the pasture. And literally the very next day, I went out and I set up these cages to exclude wildlife. So half of the cages in the bahia grass section, or half of the points of the bahia grass section had cages, and half of the points in the bare ground section had cages. And then the other points were just open to whoever might have like come by to eat the emerging seedlings. And also this whole time we were soil moisture testing and also soil temperature testing, and we did that at various points during the experimental run. In February, we did species composition, and we measured stem count of all the overseeded species and percent cover of all species found. So if we found ryegrass, we would be like, okay, there are 7 stems of ryegrass, and they cover 20% of the plot. But if we found American fern, we were only looking for the percent cover of that. So we were like, oh, American fernweed's here, 1% of the plot. We weren't counting those stems. During the first week of March, we did our second round of soil sampling. And if we look right here, this is very exciting. I made Stephen take a picture of it. You can see that this area is experiencing leaching and this area is experiencing accumulation of nutrients. I was so excited when I saw this, guys. You don't understand. And then when I got all this data back, I conducted a Shapiro test for normality. And if the data was normally distributed, I did an ANOVA test for significance. If it was significant, I did a Tukey test. But for the most part, my data was not normally distributed. So I ran a Kruskal-Wallis test for significance, and I did a Dunn test if they were significant. For those of us who need a reminder, significance is a p-value of less than 0.05. On to the results of discussion. Okay, we are going to start with germination, which is what we use seed or stem count to assess. And we're going to focus on 2 species, and then we're going to look at all of the species as a whole. So for right now, just think about radishes and oats. This is a graph. And on the x-axis, we have ground condition. So that's bahia grass versus the bare ground plots. And on the y-axis, we have the average number of daikon radish stems. The 2 different colors represent the 2 different seed mixes, because remember, the radishes were in both of them. And these bars are like standard deviation. So what we found is that ground condition did not affect germination of daikon radishes. And if we move over to the cages, we've also found that cages, which are now on the x-axis, also did not affect germination of radishes. And we see the same thing for oats, where there was no effect of ground condition or cages on their germination. And in the most nightmarish table you will ever see, we actually found that there was no significant impact of the ground condition or caged versus open when it came to stem count of these overseeded species. But let's move on. Now we're going to focus on growth and establishment, which is what we use percent cover to assess. And we're going to think about these same 2 species. What we found was that when it came to percent cover, ground condition was significantly different between the bare ground and the bahia grass. With bare ground, the radishes got bigger and covered more of the area of the plot than in the bahia grass. But the cages didn't have any effect on percent cover of daikon radishes. And with the oats, we found the opposite effect. So the ground condition didn't really seem to affect whether or not they got big and covered the area of the ground, but the cages were hugely significant on whether or not the oats like covered the ground, with there being a higher percent cover of oats in the cages than in the open plots. And when we zoom out to look at all of the species, we find that it's very species specific. With the bare ground versus bahia grass, that ground condition only really affecting the radishes and ryegrass, and the cage is only really affecting the oats. These dashes right here means there was like no variance in the data. Usually it's like there was only ever like a percent cover of 1. Okay. What does this mean? Did bahia grass hinder the overseeded species? When it came to germination, no, they seem to germinate regardless of whether or not they were in bare ground or bahia grass. But with percent cover, yes. Although it does seem to be species specific, ryegrass and radishes got much bigger and covered more of the area of the plot when we planted them in the bare ground area. And does wildlife exclusion help the overseeded species? Not really for anything, except for the oats. They did get bigger and covered more ground area in the cages. So maybe just deer are like really big fans of oats. I don't know. So if we go back to our hypothesis page, we could put a big X on number 2, and we kind of put a yes and a no on number 1. Like I said, it's pretty species specific. Okay, let's switch gears and talk about soil phosphorus. Very, very sadly, I have not received my second round of soil samples back from the University of Florida when it comes to soil phosphorus storage capacity or plant available phosphorus, but I did get water extractable phosphorus back late last week. So that's exciting. And I know that not everybody spends a lot of time thinking about soil phosphorus. So before we go any further, let's just talk about what these things mean. Soil phosphorus storage capacity, or SPSC, is the amount of phosphorus a soil can hold before it becomes an environmental risk. So basically a positive value means it's storing phosphorus, and a negative value means it's leaching phosphorus into the environment. Plant available phosphorus is exactly what it sounds like. It is phosphorus that's available to plants because it's in an inorganic form. And water extractable phosphorus, or WEP, sorry, I've got like abbreviations for everything. Water extractable phosphorus is phosphorus that's water soluble. So it's like all of the like phosphorus that's going to be released from the soil when it comes into contact with water. Okay, let's go. This is a GG plot. And on the x-axis, we have the average soil phosphorus storage capacity. And on the y-axis, we've got soil depth. And then also all of these colors represent different soil depths. Remember, the negative numbers means it's going to leach, and the positive numbers mean it's going to store phosphorus. And on the left-hand side, we have bahia grass, and on the right-hand side, we have bare ground, and they're just in the same graph. They're not like interacting with each other. I also have these nice layers over here if you're more of a visual person and want to like think about where you are in the soil profile. Okay. So we did not find any significant interactions or differences between the bahia grass and bare ground or any of the soil depths. But what is interesting is that there is not a high likelihood to store phosphorus anywhere in the soil. Like it barely breaks 10 here. And we do see a pretty like negative, like a higher likelihood to leach phosphorus in the bare ground, which we think is because of disking. We remember disking is a tillage practice, and that's chopping up everything that's in the soil. So you're breaking up plant roots, you're breaking up plant fungal associations, microbial communities, all of these wonderful things that are there that could store phosphorus we just like got rid of. So that's why we think there's this like big leaching potential here. Moving on to plant available phosphorus, again, we have the soil depths here, and if you want to look at this, feel free. We saw a significant difference in the amount of plant available phosphorus between the bahia grass and the bare ground sections. And then within the bare ground, so don't think about bahia grass right now, just think about the bare ground depths. We saw that the 15 to 60 centimeter layers were significantly different than the topsoil, which is interesting. Why do we think this is? Oh, there we go. Okay, so as you get more negative in your SPSC, your plant available phosphorus gets more positive. So we saw a big negative number here, so it makes sense that we see a big positive amount of plant available phosphorus in these same layers. And we took out all the plants. So there's nothing to take up this plant available phosphorus. So it's just like hanging out. Yeah. Okay. Here's the exciting bit. This is our water extractable phosphorus. And as you can see, we have the pre-planting data up here in the lighter color and the post-planting data down here in the darker color. We only saw a significant change between the preliminary and post-planting 0 to 15 layers of the bahia grass, and it was a reduction. We didn't see any other changes, or significant changes at least, between any of these other layers. Oh, wait, there we go. So, okay, I literally just got this data back, so I have not had a lot of time to think about this, but here's what I've got so far. It's really interesting that the bahia grass plus the overseeding mix is effective at decreasing the WEP in the topsoil, which is what you want when you're overseeding, and that's the goal of having this functional diversity of plant roots. Over the course of the overseeding project, we have seen that we have higher water extractable phosphorus in the upper layers of the soil. So this mitigation potential is actually very, very exciting. This could also be an indicator of what our change in SPSC is going to look like. SPSC and water extractable phosphorus have that same kind of relationship where when it's when SPSC is positive or around 0, the loss potential is kind of minimal, but as you get more negative with your SPSC, you see a bigger increase in water extractable phosphorus. So interesting that these are somewhat high, might not be good news, but you also might be saying, Abby, these are not very high values anyways. Remember, we're in a spodosol, which is what's it known for, guys? Nutrient leaching. Any amount of phosphorus that's up here that could be lost to the environment is going to be lost to the environment because of this high leaching potential. I wish I could tell you what number 3 meant, but get back to me. Maybe UF will get back to me too. Really quickly, some limitations. The dished area in pasture I was very heavily trafficked, so I think people might have run over some of my plants there, to be honest. And it also crossed 3 different types of soil. There was one point in an entisol, there's a bunch of points in the spodosol, and some in the histosol, which are all soil orders. Sorry, guys. Whereas the bahia grass section was only in a spodosol. We were kind of limited to where operations could disc. So gotta take what you can get sometimes. But this soil moisture gradient change actually was very interesting to look at the relationship with the overseeded species percent cover. If you're interested, I have a beautiful graph. Obviously this could be scaled up. The overseeded project at Buck Island Ranch is over 3 different pastures. I only did mine in 1, and there's also a summer mix. Surprise. But we did this in the winter, so we only did the winter mix. And my before and after data is incomplete, which is kind of a bummer. And then future research, they're going to take this into how it affects, how you could plant these mixes after sod's pulled. I think you should probably reassess some of them because we didn't see any triticale. We saw very low turnout from turnips and wheat. And maybe species that are better for Florida might be a good idea. Also looking at the below ground biodiversity relationship with the overseeded species would be super cool. And some people at Miami-Dade College are looking at that, which is awesome. I'm a product of my environment, and my environment was incredible. And I would not be here if it was not for the immense support and help and care of the agroecology team, especially Karen Rice David, who guided me through this process like a small baby bird who was learning how to fly. I'm immensely grateful. And to Greg and Bessie for hiring me, thanks. I had a great time. To everybody on the operations staff who did all of the heavy lifting and heavy machinery operating for this project, I am very grateful. To everybody on the research staff, especially those who helped with field work, thank you. And to my fellow interns for living with me and listening to me talk about radishes all the time and also helping with my field work. Thanks, guys. Questions? Hit me. Yes.
Did you eat any of the radishes?
No, I wanted to leave them in the soil, and then, well, I pulled a radish from like the old sugarcane field, and I was like, maybe I should eat this. But then I was like, didn't they spray this down? I don't know what's in the soil here. So I have a radish in my freezer at the moment, which I didn't bring because it looks kind of gross. And so no, I didn't eat, but I have had daikon radishes before, and they're like, fine. They're like nothing to write home about. Yeah. Yes.
Couple of questions about the phosphorus. How deep was the disking operation?
We think it went to about 30 centimeters. That's like what Mary Margaret told me. So yeah, 30 centimeters.
So that
give or
kind of
take.
like mix everything up in the 3 and then.
Yeah.
The second question is, after how much time after planting the overseeding species did you sample the soil for phosphorus?
About 3 months. So soil, they planted on December 6th, and I sampled on March 5th and March 6th. So it was like 3 months after. Oh, yes.
So with the soil horizons, like what's the expectation for the evolution of those with Florida? I'm thinking like in a vacuum with no water, moving your phosphorus around, how long would it take for it to go through each of those horizons?
I honestly don't know. I assume that it might, if there's like no water, it might just sit there. But yeah, I think water is the big thing that's moving it around. And I don't know how long it's gonna take to break down. So yeah.
When you were originally talking, you were talking about like legacy phosphorus and you said something about like 1980s or something like that.
Yes. So the phosphorus fertilizer was added for about 26 years before 1987. I don't know if it was every year or if it was like every other year. But it did just like leach down through the soil. I don't have like a time profile on how long phosphorus takes to leach through the soil. But then it accumulates in that spodic horizon. Usually I think because there's like a hard pan there and it's hard for things to move past that. But I have to dig into the geology. Yes, Betsy.
I was surprised by the WEP results in bare ground.
I was also surprised by that.
Do you have any thoughts about why that, like it didn't decrease after
No.
planting, but it did in the Bahia grass?
Yeah, I think in hindsight, probably should have just had a Bahia grass strip that wasn't planted at all. Maybe the Bahia grass is much more effective at mitigating the phosphorus in that topsoil than we gave it credit for. And again, maybe the disking like 3 months down the line was still having an effect. I mean, you lose all of those like roots that have been there forever, those other associations that have been there for a long time. So things are new, you know. Yes, Lucy.
So you were talking about potentially, using species that are better suited to Florida. Do you have any idea what those species might be?
No. I haven't looked into it. Yes.
So simply one of the co-benefits of this is to increase forage availability for
Yes.
cattle as well. So I wondered, and I am no doubt you're sort of stuck in the US lab mess, but I wondered, could you just give us some observations on how this is actually increasing forage as well as looking at it from mining soil phosphorus?
Yeah. So one of the reasons they do overseeding is to increase the forage production and quality. And there are species that are pretty palatable. I'm looking at Karen because she would know. And when you get a more, the problem is like we haven't seen like a lot of establishment from them, so we don't really like, I guess, know like how the cows are actually going to interact with these plants. But they're palatable and they're like going to be growing when the Bahia grass is allegedly dormant. So it could be like a boost sort of to the cows. Did that answer your question? I got sidetracked and thinking about cows.
Well, just thinking about the cost of doing it.
Yeah, and that's another big thing. It's like if these mixes aren't establishing, like as a rancher, I don't know if I would plant them because like you just have to keep planting them too. Like Ashidamene, which is one of the summer mix plants, will reseed itself, but like daikon radishes aren't going to do that. Like maybe they will, I don't know. But like for the most part, you have to replant a lot of these species, and so it might not be cost effective, especially if you're not seeing any of them come up.
Well, it's sort of interesting because these systems are classically perennial systems.
Yeah.
And it is interesting that we're, maybe we need to focus on perennials because those are the ones that really have the long-term staying power, but that's just an armchair observation.
Yeah. I like a good armchair observation. Steph, again?
You got sidetracked thinking about cows. I got sidetracked thinking about bugs. Do you think there's any potential to like do flowers in this to give like some kind of pollinator ecology?
Yeah, actually, well, the daikon radishes have the most wonderful flowers. At least I think so. And hairy vetch also has a beautiful flower. And like in the summer mix, oh my God, this thing. In the summer mix, we had another intern who did look at the amount of pollinators that are attracted to sun hemp, and found that there was a big bump in biodiversity of like butterflies and moths and things when you do plant, like it was sun hemp, so it was the summer mix, so I can't make like a direct comparison, but they saw a big bump in biodiversity. Yeah.
Yeah, so I was curious, since you weren't finding effects on phosphorus, maybe it's a function of just the species that were used and their rooting depths and how they accumulate phosphorus. I was wondering if you were to just design your own plant cover species, like what would its rooting structure and nutrients look like to actually do what you want it to do?
I would like to keep the biodrills because they do scavenge for nitrogen and phosphorus pretty well. I think like those are great. I'm big on daikon radishes. And I think the fiber, like I don't know if I have like exact examples to give you, I'll be honest.
Well, like a hypothetical plant, like is it going deeper into the soil?
Oh, yeah, it's deeper, I think deeper into the soil and just like has that ability to like scavenge and, yeah, I think like the legumes are interesting too, because they're making like nitrogen more available. So like that's a nice bump to the ecosystem. But yeah, I think they deeper and like actually growing well. A lot of the radishes we saw were pretty small, but they like have this ability to become massive. And so it would be nice if they had gotten massive, but they were all pretty much like this big when I was looking at them, but they have the ability to get like this big. Yeah. Greg.
You mentioned you were also measuring soil moisture throughout the experiment.
Yes.
My question is, did you try to compare soil moisture at the start of the experiment and at the end of the experiment? Did it change? And could that explain some of your result with water extractable phosphorus?
Yeah, actually, I didn't look at that. I did look at how, if I go, oh, yay, okay. What is this? Okay, this is the overseeded species cover and the average soil moisture from December to March. So I did look at that, but I didn't compare the soil moisture from like right after planting and then like right when we were doing the second round of soil sampling. But I should do that. That would be interesting. Yeah. I
I
don't know.
mean, this data
Yeah,
shows that you're
yeah.
aggressive.
This is the average. So like these purple points are the overseeded species percent cover in the pea grass. And the pink points are the overseeded species percent cover in the bare ground. You can see there's like a pretty long, like wide variation in the average soil moisture on the bare ground. And it gets a lot higher in the bare ground as well. Yeah, it's a nice graph.
So one of your hypotheses was not that water competition is the driving factor, and you really would get that out of the soil moisture data.
Yeah. We took it just because it's like nice data to have. And like if like the environmental conditions are the driver, like the soil moisture and temperature could be a good way to get at that. Hi, Laura.
Hi, I'm getting really tired. We have a question from Sonia.
Oh, that's my mom. Hey, Mom.
Looking back over the past 6 months and being from the north, what surprised you about Florida, and what was the environment or, sorry, was the environment and were what you expected?
She would ask me a question like that. It's really hot here. But my seasonal affective disorder wasn't that bad this year. That was nice. It was great. I loved it. Yeah, I don't know if that answered her question. I just like I heard the first part and I was like, gosh. She's asking me to write a paragraph. Sorry, Mom.
Thanks, Mom.
Thanks, Mom. Yeah, I like Florida. It's nice. I'm staying, so.
Yeah, I forgot to mention Abby's going to do her master's at University of Florida.
Maybe I'll get my soil samples there when I give them tuition.
Working on invasive plants.
Yes. Yes. Yeah. Yes, Amartya.
So let's say the radishes, they establish well, they grow big and all that. When they die, when the plants die, what would happen with the roots? I mean, they decompose and would they leave gaps in soil?
Yes. Oh, man, yeah.
So that's
They,
interesting.
the reason people use daikon radishes a lot is to break up soil compaction and because they are like these like biodrills. And when they die and decompose, they do leave these big gaps for like water and air to kind of like percolate through. And like they're like, obviously the nutrients are leaving them when they decompose, but it kind of becomes more available because it's like over the whole soil profile. Yeah. Any more questions?
I think
Or is Betsy cutting me off?
I'm cutting you off. I think we should have informal discussions outside, so thank you very much.